Hello students, let's talk about enzyme-linked immunosorbent assay or ELISA. Introduction. Enzyme-linked immunosorbent assay or ELISA is a sensitive immunotechnique used for detection of antibodies or antigens present in a given sample. It has vast application in medical diagnostics and research. The technique is based on antigen-antibody interaction. The technique uses the specificity of antibodies and the sensitivity of enzyme-catalyzed reactions to detect and quantify antigens present in a given sample. Principle. This technique is based on antigen-antibody interaction. The antigen is first immobilized onto the solid phase of the plates used for the assay or to some beads. Immobilization is achieved by coating the plates with antibodies that bind to the antigens firmly. Then antibodies specific to the antigens are added. These antibodies are linked to reporter enzymes. On addition of the substrate, the enzyme will convert it to products, and this is accompanied by change in color. Wherever the antibody binds to an antigen, the colorimetric change in color is detected. This technique is based on layer by layer addition of different molecules which link with each other. The antigen is an important molecule which is linked to both the solid phase, the plate, and to the antibody that is bound to the signal molecule. The antibody coating is also important as it concentrates the antigens present in samples, which may be sometimes very low. Hence, ELISA is a very sensitive test which can detect antigens at very low concentrations of about picomole range. Types of ELISA. There are different types of ELISA. Some of them are direct ELISA, indirect ELISA, sandwich ELISA, enhanced ELISA, and competitive ELISA. Direct ELISA. Direct ELISA is a simple plate-based method in which the surface of the plate is first coated with a sample containing antigens. A blocking protein like bovine serum albumin or BSA or casein is used to block the surface on the plate which are not bound to the antigen. Antibodies labeled with enzyme and specific for the antigens are then added. After a washing step, only antibodies that bound to the antigens remain, which is detected by the change in color of the enzyme substrate. This method is also used for identification and quantification of antibodies, proteins, etc. from a complex sample. It is a fast method as it uses only one antibody and has less number of steps. Indirect ELISA. Indirect ELISA is also a plate-based assay with two steps of antibody treatment. In this method, the plate is first treated with samples containing antigens, blocked with BSA, and the first antibody, called the primary antibody specific to the antigen, is added. Primary antibody is not labeled with enzymes. After a washing step, a second antibody that is specific to the isotypic determinants of the primary antibody is then added. This secondary antibody is labeled with enzymes and it will bind to the primary antibody. Therefore, whenever the antigen is present, primary antibodies will bind to it and the secondary antibodies will then bind to the primary antibody. Thus, on adding substrate, the enzyme will give a colored product if the antigens are present in the sample. it is a more sensitive assay compared to the direct ELISA, as more than one label antibodies is bound per one primary antibody. It is also more cost effective and flexible, as different primary antibodies could be used with the same label antibody. The main disadvantage of this technique is cross-reactivity that may increase non-specific signal and more time consuming, as it has an additional incubation step with the secondary antibody. Sandwich ELISA. In sandwich ELISA, two antibodies are used to detect the antigen of interest in a sample. One of the antibody is coated on the ELISA plate and it will capture the antigens present in a sample. The other antibody will be used for detection of the antigen. The antigens should therefore have at least two binding sites for the two antibodies. There are two types of sandwich ELISA. They are double antibody sandwich or DAS ELISA and triple antibody sandwich or TAS ELISA. In double antibody sandwich ELISA, two antibodies are used, one to capture the antigens from the sample and another for detection. In this method, plates are first coated by an antibody specific for the antigen, blocked by BSA, and the test samples containing the antigens are added. This allows binding of the antigens to the pre-coated antibodies. Then another antibody specific to the different epitopes on the antigen is added. This second antibody is labeled with enzymes which will aid in detection of the antigens with an enzyme reaction that gives colored products. In triple antibody sandwich ELISA, three antibodies are used. One antibody will be used for capturing the antigens onto the plate. The second antibody will act as a primary antibody that binds to the captured antigen, and the third antibody, which is labeled with enzyme, will act as the secondary antibody that aids in detection of the antigen. The first and the second antibodies are specific to the antigen at different epitope sites, as in double antibody sandwich ELISA. The third antibody is specific to the isotypic determinants on the second or primary antibody. Both the sandwich ELISA methods have high sensitivity, about 2 to 5 times more sensitive than the direct and indirect ELISA. The antigens used in this method need not be purified, as it will be captured onto the plates by the captured antibody. It is also highly specific, as two antibodies are used to capture and detect the antigens. These methods are also flexible, as both direct and indirect detection methods could be used. Enhanced ELISA. This is a method to enhance the sensitivity of ELISA from picomolar to a higher range. There are two major ways to increase the signal in ELISA. One is the use of multivalent molecules like the biotin-abidin system, and the other is the use of two-enzyme system. In the biotin-abidin system, both these molecules are tetravalent and it can amplify the enzyme signal. The detection antibodies are labeled with biotin and the enzyme is linked with abidin. 4 enzyme-linked abidin molecules will bind to the biotin on the detection antibody, and thus substrate conversion to colored products will be highly amplified. In the two-enzyme system, the product of the first enzyme will induce a change in the second enzyme, which will generate the signal. For example, in alkaline phosphatase or AP conjugated antibodies, AP can also catalyze conversion of NADP to NAD. The NAD produced can further be converted to NADH by a second enzyme, alcohol dehydrogenase. NADH is further converted to its oxidized form NAD, which is chromogenic and can enhance the color development. Competitive ELISA. This method is used for detection and quantification of molecules like hormones which are present in very low amount and have single epitopes. This method is different from the other methods listed above. Here, the antigen to be tested from samples is added along with another antigen conjugated with enzymes. The two antigens compete to bind to the pre-coated antibodies on the plate. If the test sample contains the antigen, less number of conjugated antigens will bind and the signals will be less on addition of the enzyme substrate. If the test sample contains less number of antigens, more number of conjugated antigens will bind and more signals will be observed. This method is also used to quantify antigens and antibodies when the assay is conducted along with a standard curve. It is also a very sensitive, specific and flexible method. Application of ELISA. ELISA is used to detect antigens or antibodies in a given sample. The antigens could be hormones, peptides, proteins, etc. It is used to detect food allergens in food industries. It is used for various diagnostic purposes like HIV, hepatitis, STD, SLE, etc. It is used in pregnancy testing. It detects the hormone human chorionic gonadotropin or HCG in urine which is higher in women that are pregnant. It is used in cancer screening in ovarian and breast cancers. It is used to test for illicit drugs like cannabinoids, amphetamines, opiates, cocaine, benzodiazepine and methadone in urine samples. It is used in disease outbreaks like bird flu, cholera, etc. It also has vast applications in research. As a conclusion, ELISA is a very sensitive and robust technique with vast applications. The technique uses the specificity of the antigen-antibody interactions and the sensitivity of enzyme reactions to detect as low as picomolar concentration of various antigens and antibodies present in a given sample. The technique is based on the immobilization of the antigens and addition of enzymes labeled antibodies specific to the antigen. On addition of the enzyme substrate, the presence of the antigen is detected by the colored product of the enzyme reaction. There are different types of ELISA with varying sensitivity and applications. ELISA has vast applications in medical diagnostics, cancer screening, illicit drug testing, research, food industries and many more. Thank you.